Hi, good evening, good morning, good afternoon, everybody, wherever you are. It is my great pleasure to welcome you all to this IACH Journal Club. This is the traditional journal club by the International Academy for Clinical Hematology. I'm Mohamed Moti from the Sorbonne University and Saint-Antoine Hospital in Paris in France. And it will be my great pleasure to moderate today this journal club. The IACH Journal Club is supported through a generous unrestricted educational grant from Takeda Oncology. And for today's journal club, the scientific steering committee of the IACH has selected this article entitled Identification of Bacteria-Derived HLA-Bound Peptides in Melanoma by Kalawara and Nagler and colleagues, just published in the journal Nature. And for this journal club, actually, as panelists, we are very proud and honored, actually, having the first co-author of the article, Dr. Adi Nagler, who is a scientist at the Weizmann Institute in Israel. And as a second panelist, it is also my pleasure and honor to welcome Dr. Martin Larsen, who is an associate professor at the Sorbonne University in Paris. So this paper represents, in my opinion, a true breakthrough in the field. And although we are in the clinical hematology arena, you will realize that, in my opinion, these findings will have some significant impact in the field of cancer in general. So you can see the abstract of the work of this work, where, to make a long story short, the authors use the 16S RNA technology, gene sequencing, and the HLA peptidomics to identify the peptide repertoire derived from intracellular bacteria. And we know that some bacteria would infiltrate the tumors. And they have identified this peptide repertoire that was presented on the HLA-1 and HLA-2 class molecules. And they have selected melanoma. They have analyzed 17 melanoma metastases derived from 9 patients. And the key findings, and you can see this on the photo that I have added to the abstract, the peptides derived from intracellular bacteria actually can be presented by the tumor cells and elicit immune reactivity. So really, this is a crucial finding, because obviously, as nicely summarized by the authors, it can provide some insight into the mechanism by which bacteria can influence activation of the immune system, but also response to therapy. And this is one nice figure from this article showing the different classes, orders, and genus of the different intratumoral bacteria that were found in the metastasis of these melanoma patients. And you can appreciate these are very important species or classes: the Bacteroides, the Clostridia, the Acinetobacteria, and many others, and we in the hematology field are actually, unfortunately, quite familiar with these bacteria because, unfortunately, our patients are really suffering from different infections. And in line with the capacity to elicit an immune activation, you have this very nice panel in the paper showing the tumor infiltrating lymphocytes reactivity towards the bacteria derived from towards the bacteria derived antigens. So clearly very important, very amazing findings that we thought, although this is not 100% in the hematology field, but you will see that it can have some implications into our field. This is why we have chosen this very important article. So my first question is going to be to Adi, if you allow me to call you Adi, but I think you authorized me in the beginning before we started the live broadcast. Can you tell us, Adi, about the rationale behind the study? Why did you decide to embark in this study? And why did you choose, for instance, melanoma and not, I don't know, breast cancer or something else? Please.
Okay, well, thank you for inviting me. I'm a PhD student in Professor Yardena Samuel's lab, and our lab studies melanoma tumors, which are known to have a high mutational load. These tumors are very interesting genetically, but also in recent years, they've been shown to be very interesting. The immune response in these tumors has been studied intensively in recent years. So this is the reason that our lab studies melanoma, and we are interested in studying the immunopeptidome of these tumors, which are the peptides that are presented on the tumor. So it can be different types of peptides, neoantigens, which are mutated peptides, or other types, and seeing if they elicit an immune response. So regarding this paper, it started as many studies have been shown in the gut microbiome regarding their effect on checkpoint inhibitors and clinical outcome in different types of tumors. And there was a recent publication in Science by Ravid Straussman that he showed that in different types of tumors, there were solid tumors, the presence of specific species of bacteria, and there was an increase in these bacteria compared to normal samples. So for these reasons, we are interested in testing if we can identify peptides on our melanoma tumors, bacterial peptides, and if these peptides will elicit an immune response by the autologous TILs from the same patient that were found inside the tumors. So this is what we tested as you summarized our paper. So we checked this in different metastases. We found peptides from both HLA class 1 and HLA class 2. We checked that the species that we identified are able to enter the melanoma cells in vitro experiments, and we showed that some of these peptides also showed T cell reactivity by secretion of interferon gamma.
So you mentioned other tumors where we know that they are quite infiltrated by different bacteria. Can you give us some examples, because obviously the audience may be interested because they have the divergent interests?
Okay, so I can just, bacteria have been shown in tumors for like over 100 years, but I'll just say the wide analysis that the paper I referred to before shows. He compared breast cancer, melanoma tumors, brain solid tumors, and they show really specific species for each type of tumor of bacteria, and this hasn't been shown before. So our paper was sort of as a result of seeing these results.
Thank you, brilliant. So Lars, you didn't participate or contribute to this work. However, you've been extremely active in this field of microbiota immune therapy for many, many years, and you made some important contributions. So what was your first reaction and your thoughts when you have seen this paper and these findings?
Okay, yeah, so thank you also to invite me for this webinar. Before I answer the question, just 2 seconds on where I'm coming from. So my interest is the interaction between host immunity and the microbiota, and we are particularly interested in secretory IgA as a regulator of microbiota homeostasis. So when I look at this study, so we are actually presently working on hematological cancers and FMT, so fecal transplants in hematological cancer patients, and so there's a lot of interest within the interest of this journal club. So what was my first thought when I saw this paper? So this is a great work. So first of all, congratulations. It's a huge amount of work, and of course they've chosen to look at metastasis. To look at the melanomas, because as was just explained, they are particularly interesting because there is this history of known bacterial invasions of this site. The interest of looking at why bacteria and cancers may interplay is obvious from the sense that the bacteria, this is kind of the domain that I'm looking at on, are directly influencing the human immune system. So even if we disregard the presentation on HLA class 1 and 2 molecules, bacteria are involved in regulating host immunity. So just from that perspective, all cancers, there is a component of microbiota influence on the host immunity that may impact the cancer itself and the therapy that we want to associate with it. However, this study is particularly interesting because it's focusing not only on how bacteria regulates immunity, but actually how specifically it can present antigens to the host immune system, thus recruiting T cells, CD8 and CD4 T cells to the tumor site, and so I think one of the big questions that I hope we're going to discuss today is what do those T cells that they actually do study in this paper, the TILs, how can they contribute either negatively or positively to the evolution of the melanomas? So if I just can finish off by saying I felt a little bit when I read this paper like the old professors in the film Theory of Everything about Stephen Hawking. For those of you that followed that or saw that movie, at one point Stephen Hawking is developing on his new theory of everything and how the universe was created in this Big Bang Theory. And the point was that most of these professors in the 60s were just leaving the room. And I'm feeling like the old professor in the room at the moment, because one of my main issues, and I hope we're going to discuss that, I'm going to stay in the room, I promise you. But one of my main issues with the paper is the fact that you see almost more, not almost, you see more class 1 presentations in these tumors. And so one of my main issues from an immunological standpoint is when you get bacteria in the site of a tumor, you would expect class 2 expression. Just by the fact that they're going to be processed and represented. This is external proteins that are coming in, being processed and presented, and this should be class 2. In a hematological context, I don't think I need to explain that more further because I think you all know. So we know that there's cross presentation going on, in particular in APCs, dendritic cells, maybe some macrophages. But here we're talking about tumors. And even though they do present class 1 and class 2 HLA molecules, I'm just really struck by this, for me, problematic issue of how to explain why you get more class 1 presentation in tumors than you get class 2. So this is a subject that I hope we're going to debate. I think it's really prone for rewriting the textbooks. which is of course always what we want to do in our field. But at the same time, as the old guy in the room, I'm going to try to defend a little bit the let's say the textbook theory, and I hope we're going to discuss how that result can occur. Yeah.
Okay, we'll dig deeper into this. Thank you very much. But I think this is very clear, and I believe, I mean, people attending this webinar, in my opinion, and myself, we are all believers in the strong interactions between the microbiota and the immune system and its positive or negative impact on disease control, I would say, in cancer. So let's dig a little bit deeper into this story, Adi. And in your paper, you have used the 16S technique. But I mean, I thought this is like a sort of a very broad, bulky way, less refined way of looking into the to identify the different bacteria species. So in another word, why did you use this, and do you believe this is the optimal way for looking into these bacteria?
Mm-hmm. Yes, so usually there's two types of, in order to identify the species, so there's two types of methods used. So it's or to do like shotgun sequencing, whole genome sequencing, or to use the 16S sequencing, which amplifies the sequences, and then you can identify them better. So when we're looking at bacteria inside these solid tumors, the amount of bacteria is very, very low. It's around 1% or even less. When we were looking at the peptides, so I can compare the peptides identified from bacteria from humans, so it was about 0.5%. And so it would be extremely hard to get to identify a large amount of species this way, and so the amplification using the 16S sequencing amplified the sequences, so we could better identify this very low amount of bacteria in the tumors, which is much different than doing sequencing when you do the fecal transplantation, when you identify the bacteria in the fecal. So it's a much higher amount, and then it's easier to detect it. So most people would go for the shotgun sequencing. So this is the reason that we use this type of sequencing. And we used usually in the 16S sequencing, so it's used with 4 different targets or 4 different primers. So we use an additional 5th primer to make it, so we can distinguish more species using this method. It was a new technique, and that was pretty much the reason.
So Martin, if you have to do the same work, would you have done and applied the same technology, or would you have done something differently?
So I'm working on, as I said, interface between IgA and microbiota. And one of the things we do is actually we sort IgA positive microbiota from stool samples, in particular from stool samples from children. So the quantity of samples is very limited, and I completely agree with Adi that when you have very low sample size, you basically need to have the amplification of 16S PCR. We even do nested PCRs to amplify when we do fact sorting. And we have a paper in press, and I published in 2015 another paper where we did the whole genome sequencing approach. And basically we could show that the limit for the whole genome sequencing approach, shotgun sequencing, is 10 to the 7th bacteria to get a robust representation of the composition of the microbiota. And it's at least 1 log 10 times lower, so around 1 million, maybe even lower for 16S. So yes, here it's obvious that 16S should be applied.
Wonderful, thank you very much. So both of you are in agreement, excellent. So Adi, I would like you to react to Martin's comment about his big surprise about this issue of class 1, which was found to be predominant versus the, whatever I would say, the traditional wisdom or common thinking that we would have expected class 2 responses. Did you have, the same level of surprise, and what are your thoughts?
So I think a lot of people actually commented regarding that the melanoma cells do express class 2. It's not known why they do, but we tested many different cell lines and all of them do express this. Melanocytes are sort of known to express class 2. So we were asked about this and we checked this thoroughly and they do express it. Regarding the amount of peptide, so the amount that we received much more class 1 peptides, I think this can be, these were the results, but it can be as a result of the technique. The class 2, the typing was less, we could receive less alleles through the typing using class 2, so I think this can be also something more technical to the technologies that we use in order to identify the peptides. And also a lot of our filtration was built also on prediction algorithms, which predict the peptides that we identify to which alleles they bind to the specific patient. So these algorithms are much more developed towards class 1 peptides, which are more commonly studied compared to class 2. So for all these reasons, I think this can be a more technical issue, and I cannot say that it is like I believe that it's totally as a result of the cells will express less class 2 peptides.
One of
Martin,
the questions I actually had for that is,
are you happy with these explanations?
if I can ask a question very specifically to that, because one of the things that you do in the paper is that you sort, you purify HLA class 1 and then the HLA class 2 molecule from, so this was actually a work that I also did in my postdoc many years ago. So I know that it's really hard to do this because these are membrane proteins. So one have to realize to get these membrane proteins out is really a tough job. So you need to add some detergents to generate the membrane and then purify the individual proteins. And you want to have class 1 and class 2 apart.
Martin, she's brilliant. Of course, she did the hard job. She's brilliant. She did.
Yeah, I know.
PhD on a Nature paper. Come on, nothing new. She's brilliant.
I completely agree. So my question was not that. My question is, did you ever try to reverse the order? Try to purify the class 2 first before the class 1? And would that affect the numbers? Because I was thinking that maybe you're just losing your peptides because you go through a serial purification process.
So, okay, we have never tried to reverse it, but I don't think this is the issue. The issue is more in the filtration steps after identifying the peptides from the mass spec, and that after the analysis. So because of that, these algorithms are not so sufficient. So we tended to filter higher filtration steps for the class 2 peptides.
Absolutely.
So
okay, wonderful. So time is running, guys. So I'd like to move now to the practical implications. Of course, we can debate the techniques and their limitations, but obviously you have to live with the amount of material you have, and it's not very easy to work on human material, you know. I really appreciate the hard work and the effort you have put in this. So Martin, as far as I know, and I'm really the naive person in this discussion, bacterial antigens are non-self. So in theory, these are really very attractive targets that can be used for immune therapy and in a positive manner. On the other hand, I am usually very suspicious when it comes to cancers, and I would say, well, maybe it's a trick to evade the immune system. So exactly.
I love the question. Sorry, yeah, finish.
So exactly. So how would you? What is your reaction here? What are your thoughts? How can we build upon this information to either positively stimulate the immune system or try to block the immunosuppressive impact?
So if I had the answer, of course, I would be creating a company tomorrow or writing a better paper.
And you'll get the
myself.
Nobel Prize.
But yeah, or something like that. But no, let me just sum that up to what I think from that perspective is that we have to think about what commensals actually do. These are not. We have to understand that these bacteria that are found are not pathogenic bacteria. These are commensal bacteria that are living on our skin and are invading melanomas on the skin. And we see similar things in other tumors that are also exposed to environmental bacteria or commensal bacteria. But these are not pathogens. Pathogens, there'll be no issue because they'll clearly create an immune response that would be deleterious for the cancer. But the fact that they're commensals makes a lot of questions occur because when we have commensal bacteria in our gut, for example, which is the most colonized organ in our body, these bacteria will be processed in different manners. They'll be processed by dendritic cells in our Peyer's patches in the ileum, and they're going to be presented. And probably, and that will be another study, and I would love to see that study, doing peptidomics on dendritic cells from the domes of the ileum, of the Peyer's patches, but probably they'll be presenting the same type of peptides. And so the point is that commensals are actually generating immune responses that are considered tolerogenic, in the sense that, for example, the IgA antibody response that we are studying is considered a tolerogenic antibody response. It doesn't activate complement, and basically it's there. That's at least our working hypothesis at the same time to protect the host and the bacteria and promote colonization, good colonization. And good is a very good question how to define, but there to promote colonization that is beneficial for the host. So I would say that there is a real risk that those peptides would, rather than being positive for cancer therapy, might actually be more promoting a tolerogenic state, an innovation step. And one of the things we could maybe ask to Adi is that they did the TIL responses, they did interferon gamma responses. I'm quite surprised to see how low the interferon gamma reactivity is. Did you do PD-1 as well? Are these TILs really immunosuppressed? Is that why there's so little interferon gamma?
We didn't do PD-1, but we checked other activation markers. We did CD69 and another few that we were requested, and they were still, like, they were twofold larger than the control,
right?
Which is
okay.
considered a response.
Okay.
So I guess the answer is we don't know, huh? But
yeah,
there's work to be done
but I
there.
think, Martin, your point is well taken, because I thought the conclusion of the manuscript is very positive, that we're going to exploit all of these findings in a very positive manner. But I think we have to acknowledge the possibility of immunosuppression and immune evasion. So, guys, in the last 5 minutes of this journal club, I would like to push it a little bit further beyond the field of melanoma, maybe. And I'll start with you, Martin. Where do you believe we should, which kind of tumors, for instance, and of course, I would be interested on your thoughts about hematologic cancers, where you would try to reproduce or to do similar work? Is it lymphoma? How would you do it, for instance, in acute leukemia, where your tumor is circulating in the blood? And I've always been taught that the blood is sterile, so you're not supposed to find bacteria there. So yeah. I'd love to hear from you.
So there are quite a few papers coming out on what they called bacteremia, and then for a while it was debated whether it's really bacteremia. And at one point there came a new terminology which was called Dnaemia, which basically was that probably those bacteria found in the blood were not bacteria, but rather bacteria that had been killed by the complement, and we just had like DNA, maybe some remains of the cell, but no living bacteria. I actually lost my associate professor position for the first time I went, because one person didn't agree with me that we should not look in. They thought we should look in the blood, and my answer was, for me, a bacteria in the blood is what we call a sepsis. So I thought that was a pretty ridiculous idea. So I still stick with my idea, although I lost my position back then, that I don't think that we'll find living bacteria in the blood. Some debate that. The tumors that are having bacteria makes sense, because most of them are either, if it's a colonic tumor, they will be exposed to the bacteria of the colon. If it's a melanoma, they'll be exposed to the microbiome of the skin. So all of those make sense. I, for example, think that a breast tumor would probably not be the right place to look for bacteria, because it'll be much less exposed, to my opinion, to bacteria. But I think it's something that is still being developed. But I think for hematological tumors, I'm much more in favor of what we are studying in my team at the moment, which is whether fecal transplant can help recolonize the gut after antibiotic treatment in these individuals to modify their immune system, to give a better immune response to the tumor, against the tumor. But that's just my point of view. I'm looking forward to hear what Adi has to say about that same question.
But then you mentioned your opinion about the blood. So for instance, if we consider lymphoma, for instance, where you have like big tumors in close vicinity to the skin, would this something where you would be more interested?
Anything where you can approach it, I think physically you can argue, but even a lymphoma for me is, you can have metastasis, sure, but even the lymphoma would you consider that really being close to the skin as such?
I don't know.
Under the skin, but yeah,
I don't know. The problem is when you manipulate this, you also contaminate all of this material. So, Adi, now you've published this fantastic Nature paper, and again, congratulations. You should really be very proud of this achievement, because I don't meet personally every day with PhD students who publish a Nature paper. So this is really amazing. So if you have to continue this work and I can bring you a $1 million grant, what would be the next steps? What kind of questions, follow-up questions you would like to ask and, continue your work?
So I think like an extremely interesting process that ongoes with these tumors is that they undergo immunoediting and acquire resistant mechanisms and changes in the antigens that are presented on these cells. And as I said before, so melanoma has much mutations, and neoantigens in melanoma have been intensely studied and searched for. And there's also different types of antigens. We found bacterial antigens, but there's known to be shared antigens and overexpressed antigens and different tumor-associated antigens. So I think it can be very interesting to see the effect of, I would take a cohort of patients and identify the antigens that are presented in their tumors and try to perform cancer vaccines or peptide vaccines, but really focusing on the question, is there an effect of injection of at different time points, the different types of antigens we have, and in what order would we get a better response for these patients? And also we can see if combinations of different types of antigens would be more beneficial, or to first give only a neoantigen treatment, and then after try to combine the different presented antigens.
Well, this is really a fantastic research program. I hope everybody has enjoyed this journal club. It has been a pleasure having both of you with us. I do apologize for my ignorance. Maybe I wasn't always accurate, but this is not really my field. But I learned a lot from you guys today. Thank you again, all of you, wherever you are, for joining the Journal Club today. And I hope that you will continue to stay safe and keep well. And we shall meet in 15 days for another Journal Club. In the meantime, Thank you very much and goodbye. Bye-bye.
Thank you. Bye.